We have a number of introgenic interactions so far we have studied. For example, the codominance, in-couple dominance, the lethal genes, pleiotropy, etc. Now the next one, under the intragenic interactions, we have polygenic inheritance. What's the meaning for that one? I mentioned already, we have three different types of inheritance. One, monogenic inheritance. One gene, one character. Number two, just pleiotropy. Here a single gene controls many characters. Now third one, we have polygenic inheritance or cumulative factor hypothesis. Here a single character is affected by many genes. So many genes are involved in controlling a single trait, and that is called polygenic inheritance. So a single trait is affected by many genes. Now what's the meaning for polygenic inheritance? So many genes are combined together to control the inheritance of a characteristic of an organism. A characteristic of an organism is called polygenic inheritance. Now the polygenic inheritance was first demonstrated by a person by name Nielsen Elling. In 1907. He demonstrated the polygenic inheritance in white kernel color. We have different shades of kernel color. Actually, in the case of that polygenic inheritance. Normally, polygenic inheritance explains the continuous traits, inheritance of continuous traits, which are compatible, which are not normally equal to, not matching to the Mendelian inheritance. So, here, actually what's happening, it explains the inheritance of characters. Many genes are involved in explaining the character. Same way it explains the inheritance of continuous traits. Explains the inheritance of continuous traits, or we can say continuous variations, which are compatible with the Mendelian inheritance. Compatible with the meaning for that one, not comparable, not matchable to Mendelian inheritance. Because in Mendelian inheritance, one gene, one character. But here you see that one, we have a gradation in the characters, that is called continuous variation, what is studied. And all these normally the characters are controlled by the effect of many genes. So it is an example for quantitative inheritance. The character will be decided by the number of genes, not by the quality of the genes. So he took the wheat kernel color to just demonstrate the polygenic inheritance. The wheat kernel color is controlled by two dominant genes for red color. Each gene has two alleles. Let us consider gene R1, then R2. These are the two genes. Now it has its own allele, that is a recessive allele. And it also has its own allele, the recessive allele R2. As we said, the kernel color is controlled by actually two genes, and each gene has two alleles. So these two are the alleles and these two are the alleles. That's the meaning for that one. Now he crossed. Actually the red gene is dominant over white. Now these two genes are responsible for white color. And these two genes are responsible for red color. And each one has its own allele. Capital R1 has its own allele small r1. Capital R2 has its own allele small r2. So this is naturally the basic genes responsible for, that is the color of the kernel in wheat. Now he crossed two pure breeding varieties. So the parental generation, as I mentioned earlier, now the wheat kernel color is controlled by two genes, R1 and R2. And each one has actually two alleles. Now R1 has its allele, so two alleles. Now R2 has its own allele, that is R2. Recessive. Now just Nielsen crossed homozygous dark red, where we have 4 dominant genes, with the homozygous white, which has 4 recessive genes, that's why it's white. A crossed and obtained in the F1 generation all heterozygous medium red colored individuals with the 2 dominant genes. We have this is 1 dominant gene and 2 dominant genes. So the 2 dominant genes together produce medium red. Now the intensity of red color is normally controlled or determined by the number of origins present in an individual. The intensity of red color is determined by the number of origins. Generally I am saying that one, number of origins present in that individual. Here there are 4 dominant genes and here only 2 dominant genes. So here I mentioned already it's an example for quantitative inheritance. So the color or the character is decided by a number of genes. Accordingly we have the character or the trait. That's why I said in the F1 generation the color of the individual is determined by the number of origins present in it. Now this origin is normally having an additive effect to produce what is called the red kernel color or the color of the that is kernel in that plant. So it is having an additive effect, what is called a cumulative effect, additive effect or cumulative effect. So this is one, the F1 generation. Now what are the gametes produced? In any dihybrid cross, the heterozygous F1, which is called a dihybrid, produces only 4 different types of gametes. Here I am taking, as in the case of other cases also, taking the first 2 letters, capital R1 twice, small r1 twice. Taking the next 2 letters alternately, let us see that 1 or 2. R2, capital R2, then small r2, capital R2, small r2. So this is a way you can find the number of gametes. Taking the first 2 letters, writing twice each. Taking the next 2 letters, that is R1, writing twice each. And adding the next 2 letters alternately, capital R2, small r2, capital R2, small r2. So we are giving 4 types of gametes. In any dihybrid, though the phenotypic ratio or the genotypic ratio are changing, here the nature of the gametes produced will always be the same, 4 types, because we are taking actually 2 different characters. Now it is recrossed with another one, just what we have, the genotype R1, small r1, that is R2. And taking that is R2 dominant gene, R2 recessive gene, R2 the dominant gene, R2 the recessive gene. So when cross is effected between what we have the F1 individuals, that's called the self-fertilization, we have received the following phenotypes and also the genotypes. And how far the character is controlled by the number of genes we also see. So I want to represent in the form of a checkerboard. Okay, let's see that one. The F1 generation produced a heterozygous individual having 2 dominant genes. So it is medium red in color. When it forms gametes, there are 4 different types of gametes. In any dihybrid cross, 4 types of gametes will be formed. I mentioned already, for just finding out the gametes, take the first 2 letters, R1, capital R1, small r1, writing each twice. I am writing capital R1 twice, small r1 twice. Then writing the next 2 letters, capital R2, small r2, alternately, capital R2, small r2, capital R2, small r2. This is a way to find the nature of the gametes. So 4 gametes are produced on either side of the male and female. So random combination results in the formation of 16 individuals. Now what are the 16 individuals produced? Now as in the case of dihybrid cross, here it is not based on the quality of the genes. but depends on the number of dominant genes. More the number of dominant genes, more will be the red color. That's a dark red. Then we have medium dark red, or medium red, or light red, or simply white. So that is being decided by the number of genes, hence called quantitative inheritance. Now the random combination, let's result now. This is capital R1, R1, R2, R2. So there are 4 dominant genes here. So R1 twice, R2 twice. This is actually 4 dominant genes. I will take the next one. Capital R1 twice, then capital R2 and also small r2. So 3 dominant genes. Let us take another one, the next case. See capital R1, that is small r1, then capital R2. See that one also. So we are writing, actually I mentioned just here, R1, that is R2 twice. So the same number what we have, that is what genotype we have here, also here in the second and third box. This is box number 1 I mentioned, box number 2, box number 3, box number 4. This is 2, box number 3 and box number 4. So whatever you have here, the same one you are getting here. So I am writing the same genotype, capital R1, R1, R2, then small r2. And in the second case we see that one I am writing capital R1, small r1, just R2, R2. This is about what we have. Then in the fourth case, if you are making a cross, then what we are getting actually, that is capital R1, small r1, capital R2, small r2. So capital R1, small r1, capital R2, small r2. The same you have in these 4. So capital R1, small r1, R2, small r2. Here capital R1, small r1, then capital R2, small r2. And here also the same one, capital R1, small r1, capital R2, small r2. Now you can find here there are 4 dominant genes. Here 3 dominant genes of one genotype. Here 3 dominant genes of another genotype. Here 2 dominant genes. Okay, so this is what we have. Now if you are taking the next one, I am writing here R1, another R1, then R2, R2. So 2 dominant genes as here. Okay, then I am writing here R1. Just see that one, I am writing R1, then R1, then R2, R2. So the same one, but we have only 1 dominant genes. We are getting the same one here also. As in the case of monophysaridae cross, remember those things also. See, in these cases, you see that one, 2 dominant genes here, 1 dominant gene, 1 dominant gene. Okay, so we are getting actually in this case 4 dominant genes, 3 dominant genes of one genotype, 3 dominant genes of one genotype, and 2 dominant genes of one genotype. And also we have 2 dominant genes of another type, just like that one. And we have to go for the cross here once again. So what we have, R1, R1, see, then R2, R2. So here also we are getting what is called 2 dominant genes. Here R1, but here R2. Then what about the remaining 3? Now we have to take this one, R1, R1. So I am taking just a red color for just a difference. R1, R1, R2, R2. So R1, R1. Then we have dominant letter R2, then recessive letter R2. We are getting the same one here too. That is, we see that one, R1, there also R1. Then we have there is the next one, see, we are crossing this one, R2, R2. The same one what I am getting here. So if you want to check whether the letters are correct or not, you can go for it and tell you later now, as in the case of direct cross. These two are similar. And this one is actually having two dominant genes, but different from this one and this one. Here we have only one dominant gene. So let's go for the last one. We have all the recessive genes. We have all the recessive genes. Small r1, r2, r2. All recessive genes. So this is actually the receptor, but the phenotypic ratio is different. Now if we have 4 dominant genes, then we have the following results. I mentioned already, the intensity of red color is decided by or determined by the number of odd genes. Suppose we have 4 dominant genes. 4 dominant genes. What is the nature of the phenotype? 4 dominant genes. That is dark red. Dark red. How many dark red individuals are present? Only one. So only one. Dark red one. Now this is the only one dark red. There are 4 dominant genes because it is a quantitative inheritance, depends on the number of genes. Then we have medium dark red. Medium dark red. The second phenotype. Medium dark red. Now these individuals have 3 dominant genes. The number of dominant genes actually 3. Now in the dark red, the number of dominant genes 4. I am writing first the dominant genes. Now also I am writing within brackets, this is number 1, total number of phenotypes. 4 dominant genes, 1 phenotype. Then the next one, medium dark red, 3 dominant genes with the 2 genotypes. 1 genotype, what we have here, capital R1, capital R1, capital R2, that is what we have, small r2. And this one is 2 in number. Then the second one, capital R1, that is small r1, r2, r2. This is the second case. This is 2 in number, totally 4 individuals. Now the medium dark red, just actually see that one, dark red, that is 4, medium dark red, that is 4, but the number of dominant genes 3 within brackets. This number indicates actually the total genotypes, and the one within the bracket denotes the number of dominant genes. Now I am taking this one, these 2. So these are similar types and these 2 are similar types. But the total number of genes there you have only just 3 dominant genes. So totally 4 individuals with the 2 different genotypes, but the number of dominant genes 3. Let us go for this. This is number 1, this is number 2, this is what is called actually red. And this one what we have? 2 dominant genes. Red we have 2 dominant genes, but the number of individuals totally we have 6. Now these 4 and these 2. So totally we have 3 dominant genotypes. This is one genotype, 4, this is another genotype, 1, this is another genotype. So medium dark red, now the red color having what is called 2 dominant genes. I am writing the genotype. Genotype number 1, R1, R1, R2, R2. How many? Just we have 4 numbers. Okay, so dark red, medium dark red, then we have red, or we can say the intermediate color, we can say either way. Red, that is what is called intermediate red. Then the second genotype, R1, R1, R2, R2. This is 2 in it. You see that one, sorry, made a mistake, that's 1. Totally 6. Now this is one genotype and what I mentioned here. Now another genotype, this is another genotype. That is what we have, recessive genes, homozygous, dominant genes are 2, also homozygous 1. So the total number of individuals 6. So one dark red, another one medium dark red, the third one red or intermediate color is 6. Then how many genes are present in that case? There are 2 genes. Now we have 1 gene, 3 genes, and here 2 genes. So when you have 4 genes, now we are getting dark red. When you are getting just actually 3 genes, or genes, then the medium dark red. If you have only 2 just dominant genes, you see that one, here 1, 1, 1, 1, 1, 1. So then it is an intermediate red color. Intermediate red color with the 3 genotypes. Now the next one, we have also, that is light. The next one, light. Total number of light individuals also 2. Total number of light individuals also 2. Now here just the total number of individuals 2 plus 2, 4. Now the total number of individuals I am writing 2 and also plus 2 here. So I am writing totally just 4. Now these 2 are one variety and these 2 are one variety. So now totally there are 4 individuals with the light red color, each one with only one dominant gene. So now this is 1, this is another one. So 2 plus 2, one variety